afternoon, everyone. Today, I'm excited to talk about CRISPR-Cas9, a revolutionary gene editing tool that has opened up new possibilities in a variety of fields. In my presentation, I will cover two main topics: how CRISPR-Cas9 works and its application. So before I start to explain the CRISPR-Cas9 technology, I want to show you this video.
CRISPR-Cas9 is a novel molecular tool modified to allow precise gene editing. CRISPR, often referred to as the gene scissor, can cause targeted double-strand breaks in a specific sequence of the genome. What are the main components of CRISPR-Cas9? First, there is the Cas9 protein, which can unwind the DNA and has also two domains with a nuclease activity. Hence, it can cleave the DNA double-strand. The second component is made up by RNA, the tracer RNA, which is bound to the Cas9 protein, and the CRISPR RNA, which is specifically designed to hybridize with the region of interest, are covalently fused together, and this is called guide RNA. The CRISPR-Cas complex searches for the sequence of interest until the guide RNA hybridizes with the DNA. When there is a PAM sequence downstream of the target region, the nuclease domains become active and create cuts. That results in a double-strand break, which is repaired in an error-prone manner, causing mutations and most likely also the gene knockout.
Yeah, let's begin with how CRISPR-Cas9 works. As its core, CRISPR-Cas9 is a tool that allows scientists to make precise changes in the DNA sequence. The tool is made up of two main components, the Cas9 protein and guide RNA. The Cas9 protein acts as a pair of molecular scissors that can cut DNA, while the guide RNA acts as a guide that leads Cas9 to specific location on the DNA strand. Once Cas9 is in place, it can cut the DNA strands, creating a break in a genetic code. The break in the DNA can then be repaired in a few ways. In some cases, the cell's natural repair mechanism will fix the breaks, leaving the DNA strands unchanged. However, in other cases, researchers can introduce a new piece of DNA, which is then used to replace the section that was cut out. By doing this, scientists can add new genes and remove problematic genes, or even replace entire sections of the genome. Now let's discuss the application of CRISPR-Cas9 technology. The potential uses of this technology are vast and far-reaching, with implications in medicine, agriculture, biotechnology, and more. One of the most exciting applications of CRISPR-Cas9 is in medicine. By using the tool to edit genes, scientists are exploring new treatments for genetic disorders, such as sickle cell anemia, cystic fibrosis, and Huntington disease. In some cases, researchers have even been able to use CRISPR-Cas9 to edit genes in living organisms, including humans. For example, in 2020, scientists successfully used the technology to cure a rare genetic disorder called autosomal dominant hypercholesterolemia in a patient. In addition to treating genetic disorder, CRISPR-Cas9 has also shown potential fighting cancer by cutting out specific genes that cause cancerous growth. Scientists are hoping to create new therapies that can target cancer cells while leaving healthy cells untouched. Another application of CRISPR-Cas9 is agriculture. The tool can be used to create crops that are more resistant to pests and disease, which could help to reduce use of harmful pesticides. Additionally, scientists are exploring ways to use CRISPR-Cas9 to make crops more nutritious or to extend their shelf life. Finally, CRISPR-Cas9 has implication for biotechnology. Scientists are exploring ways to use the tool to create new antibiotics that can fight antibiotic-resistant bacteria. Additionally, researchers are using the technology to create biofuels from algae, which could help to reduce our dependence on fossil fuels. It is important to note that there are ethical considerations around the use of CRISPR-Cas9, particularly in human germline editing. While the technology offers many exciting possibilities, we must ensure that it is used responsibly and ethically. As with any new technology, there are risks and uncertainties that must carefully consider. In conclusion, CRISPR-Cas9 is a powerful gene editing tool that has potential to revolutionize a variety of fields. Its precise nature and versatility make it an exciting new tool for scientists and researchers. However, we must use it responsibly and ethically to ensure that it is used safely and effectively. Thank you for listening. If you have any questions or comments, I would be happy to address them. But before I end my presentation, I'd like to leave you with some interesting facts about CRISPR-Cas9. Did you know about CRISPR-Cas9 technology was first discovered in 1987 by Yoshizumi Ishino and his college in Japan. But it wasn't until 2020 that Jennifer Doudna and Emmanuel Carpentier discovered how to use it as a genetic editing tool. Additionally, the CRISPR-Cas9 system has been adapted from a natural defense mechanism used by bacteria to protect themselves from viruses, and its widespread use has led to its being called the molecular scissors of the genomic world. In conclusion, CRISPR-Cas9 is an incredible technology that has the potential to make significant advancements in medicine, agriculture, and biotechnology, and many other fields. Its precise nature and versatility make it a valuable tool for researchers and scientists to explore new possibilities. However, we must use this technology responsibly, ethically, and with caution as with any powerful new tool to ensure that it is used safely and effectively. Thank you for your attention, and these are my references.